Polyembryony. It is a phenomenon of development of more than one embryo in one ovule, seed or fertilized ovum. In plants, this phenomenon was first reported by Antony von Leeuwenhoek in 1719 in orange seeds. In a seed with many embryos, usually one embryo matures and the rest degenerate during the course of development. An embryo is formed from the repeated division of a zygote. A zygote is formed by a process of fusion of male gamete with an egg cell inside the embryo sac. Classification of an embryo. Ernst and Schnarf classified polyembryony into two types, namely true polyembryony and the second one is false polyembryony. True polyembryony may be subdivided into two types, namely cleavage polyembryony and adventive polyembryony. True polyembryony. In this, the production of embryo within or by projecting into a single embryo sac is termed as the true polyembryony. Cleavage polyembryony. Here, the embryo arises within an embryo sac either by a cleavage of egg, that is a fertilized egg, or from synergids or antipodals or the endosperm. The antipodals are present towards the collageal region in the embryo sac. Cleavage polyembryony is common in gymnosperms, but it is of rare occurrence in angiosperms. In Erythronium americanum, the first division of zygote is normal. From embryonic mass, many cells at distal end form separate embryos. In Exocarpus and Isotoma, additional embryos are formed from suspensor cells of proembryo. Adventitious polyembryony. When the embryos arise from tissue outside the embryo sac, the cells of the nucellus, but generally they come or the integument to lie within the embryo sac. False polyembryony. This type includes the cases in which more embryos are formed as a result of two or of the development of aposporic embryo sac. Embryos from cells of embryo sac other than the egg. The embryos may appear from synergids and antipodal cells in the embryo sac. The synergids, which is present in the embryo sac towards the micropylar region, may be fertilized by sperms from an additional pollen tube or develop without such fusion. In Argemone mexicana and Phaseolus vulgaris, additional embryos may appear from unfertilized synergids and hence it is haploid in nature. Embryos from antipodal cells develop less frequently. These antipodal cells are present in the embryo sac towards the collageal region. The best example for this is Ulmus americana and Allium odorum. All the antipodal embryos may not remain viable. Embryo from endosperm. Endosperm is formed by fusion of the two antipodal cells with the male gamete, which is present towards the central region of the embryo sac. It is known as triple fusion, and the product is known as primary endosperm nucleus, which develops into an endosperm, and it is a nutritive tissue for the developing embryo. So, embryos from endosperm. Embryos developing from endosperm have been reported in Balanophora and Ulmus. However, Ernst in 1913 found that such embryos developed from egg got embedded in cellular endosperm. Spontaneous and induced polyembryony. Polyembryony may be spontaneous, which includes instances of naturally occurring polyembryony. Induced polyembryony include instances of experimentally used cases. Yaclover has distinguished two types of spontaneous polyembryony, that is gametophytic and sporophytic. Coming to the gametophytic one, this arises from gametic cells of embryo sac. In case of sporophytic, these arise from zygote, proembryo, or initial sporophytic cells of the ovule. It can be nucellus or integuments. Embryo development can also be made in culture media, that is known as induced polyembryony. The embryos developed in culture medium are known as adventitious embryos, somatic embryo, supernumerary embryo, or embryoids. The causes of polyembryony. There are various theories that are put forward to explain the process of polyembryony. Coming to the first one, necrohormone theory. Here, the cells witnesses nucellus degeneration in order to give rise to a stimulus for adjacent cells to undergo division. It leads to the formation of adventive embryos. Hybridization theory. According to this theory, hybridization process leads to recombination of genes where a single unit is formed, which creates multiple embryos. Practical value or application of polyembryony. Nucellular adventive polyembryony is of great significance in horticulture. Another practical value or importance is the adventive embryos provide uniform seedlings of parental type. Next value is the nucellular seedlings of citrus provides better clones than cuttings. Another important value is the cuttings form lateral roots and the nucellular seedlings develop taproots, which is better root system. The nucellular seedlings show restoration of vigor. Moreover, the nucellular embryos are free from disease. Thank you.